and to all of our panelists for handling upwards of 40 questions that we received during this session. So thank you so much for being involved in this Q&A panel. And we will now switch gears to the second session for the day. And it is my pleasure to introduce Dr. Tannaz Ramezanli. Dr. Ramezanli is a senior pharmacologist within the Office of Research and Standards at the Office of Generic Drugs. She specializes in topical and transdermal products and is a senior reviewer for development of product-specific guidances, responding to control correspondences, citizens' petitions, and pre-NDA meeting packages. She also leads several regulatory science research initiatives related to the development of efficient bioequivalence approaches for complex topical products that are funded through the GADUFA Regulatory Science and Research Program. Tannaz received her PhD in pharmaceutical sciences from Rutgers University and her PharmD from Tehran University before joining the FDA. Please join me in welcoming Dr. Ramezanli.
Hi everyone, welcome to session 2 of today's webinar. My name is Tannaz Ramezanli and I'm Senior Pharmacologist within Division of Therapeutic Performance 1, Office of Research and Standards. And today I'm going to talk to you about practical considerations for IVRT studies with topical drug products that are submitted in ANDAs. The learning objectives of my today's talk are first, to describe considerations for IVRT study design and validation when used as a component of characterization-based bioequivalent approaches and submitted in ANDAs, and also provide clarifications related to IVRT best practices and common questions. So IVRT study can be done for multiple purposes for topical products, quality control or post-approval changes or demonstration of BE. So some of these best practices or principles that are discussed in this presentation may be applicable to all these different types of IVRTs, but the focus of this presentation is for those IVRT studies that are conducted for demonstration of bioequivalence. IVRT is a compendial performance test to study the arrangement of matter, and in characterization-based approach, IVRT is considered an in vitro bioequivalence study. What I will discuss in this presentation is IVRT in the context of vertical diffusion cell, and in this setup, a porous membrane is applied on the diffusion cell, which contains a receptor solution, and the formulation is applied on top of the membrane, and the drug that is released from the formulation into the membrane and gets into the receptor solution can be measured by sampling the receptor solution. When IVRT is conducted for a demonstration of bioequivalence, there are three major phases. One is IVRT method development, IVRT method validation, and finally IVRT pivotal study. So the first step is IVRT method development, which is exploratory in nature and is done to select and optimize the method parameters that you're going to use for a method IVRT validation and the pivotal study. So while the studies that are conducted under method development can be analyzed using an analytical method that may not be validated because you can develop your IVRT method and validate your analytical method in parallel. However, you need to make sure to report in your ANDA which IVRT studies were done using a validated analytical method and which ones were not. So for selecting method parameters, we would recommend this sequence in your method development. First is to select the equipment and the acceptable equipments used for the IVRT studies are discussed comprehensively in USP Chapter 1724. And as I mentioned, vertical diffusion cells are the most commonly used equipment for IVRT. The next parameter that you need to select for the IVRT is the receptor solution, and we recommend that you select this very early on during your method development, because you're validating your analytical method based on your receptor solution matrix. So if you select and optimize it early on, you don't need to later go back and revalidate or redevelop your analytical method. After selecting your receptor solution, then you can select the membrane and other method parameters such as dose amount, sampling times, steering rate as applicable, etc. After method development comes method validation. So as part of IVRT validation, you need to qualify important parameters and components of your method. And one of them is the equipment. So as part of equipment qualification, one important aspect is to provide empirical measurements of dimensions of the orifice or volume of the receptor compartment for each individual diffusion cell that is used in the IVRT study. And as you know, you need to use this empirical measurements value for calculating of the drug release. And if there are manufacturer information about these dimensions, you need to submit that along with your empirical measurements. Another important part of equipment qualification is to make sure that the equipment can control the diffusion cell thermoregulation, meaning that it can maintain and control diffusion cell temperature at a certain degree of your interest. And for the IVRT study, we are targeting or measuring membrane surface temperature. This is the temperature that we are interested in. And if you're evaluating a drug release from a topical product, this temperature should be adjusted to 32 degree plus minus 1. And if you're evaluating release from a vaginal or rectal product, instead of 32 degree, you will target 37. And it is very important that your equipment can stabilize and maintain this temperature throughout the study, and you can verify that by a device like an infrared thermometer and measure and monitor the membrane surface temperature. One important method parameter that you need to qualify during IVRT is the receptor solution. For qualifying your receptor solution, you need to first make sure that the drug solubility in the receptor solution of interest exceeds the highest sample concentration in the IVRT, ideally by an order of magnitude. You don't need to necessarily measure the saturated solubility or maximum drug solubility in the receptor solution, as long as you show that the drug solubility is a few fold higher than the highest sample concentration. That would be sufficient. Another aspect of qualifying the receptor solution is to demonstrate that the drug in the receptor solution is stable for the duration of the IVRT and in the condition that mimics the IVRT conditions, meaning that for example, when you run the IVRT study, if the membrane surface temperature is 32 degree, probably the temperature of your receptor solution is slightly higher, for example, 33 or 34, and that should be the temperature in which you measure the drug or evaluate the drug stability in the receptor solution. And last but not least is to establish acceptable linearity and precision of the drug release profile in the IVRT, and by acceptable linearity we mean the R-square value of equal or more than 0.97. And in the coming slides I will describe more why we recommend a higher value than 0.9. Next parameter that needs to be qualified during IVRT validation is the membrane. So membranes that are used in the IVRT study has effective pore size of usually average 0.45 micrometer. if you're using a membrane that has a larger pore size, let's say 1 micrometer or more, you need to justify why such large pore size was needed for your IVRT study. As part of membrane qualification, you need to make sure the membrane is inert and evaluate drug binding to the membrane at a concentration that is relevant to the drug concentration in your IVRT study. And it doesn't need to necessarily be the highest concentration. It should be within the range of the drug concentration in the IVRT study. And making sure that your membrane is chemically compatible with the receptor solution, and when you run this IVRT, you get an acceptable precision and linearity of the release profile with R-square value of equal or more than 0.97. You also need to qualify the receptor solution sampling and making sure that it is accurate and precise in collecting receptor samples. And we would recommend for you to use a sampling technique that can reliably collect a consistent volume of the sample from a well-mixed volume of the receptor compartment. For example, if you're using a vertical diffusion cell with a sampling arm, if you just sample from the arm, you may not get a sample from a well-mixed volume. So we recommend that you make sure you sample from the center of the receptor compartment. Also, we recommend that you submit manufacturer's specification for the accuracy and precision of receptor solution sampling if such exists. That is especially recommended when you use automated sampling technique with your equipment. Now, you're qualifying your receptor solution sampling, but you also need to make sure the frequency of sampling and the number of sampling time points are adequate. So for the IVRT study, we recommend at least 5 sampling time points, and you need to make sure those sampling time points are selected at the time where you're at steady state release kinetics. For example, here, this is a release profile. But if you look at the first time point, the release rate at this time point is different than the release rate of the rest of the profile, and here we are not at steady state yet. So it is not valid to use this time point to calculate the drug release rate. So what we recommend here is just to exclude this time point, and in this case you have 5 out of 5 sampling time points left, so it should be okay and acceptable to just use this. So even though the R-square value was 0.98, unless you really plot the data, you wouldn't see that the first time point is deviating from steady state. So making sure that all the sampling time points that are selected are at the steady state release. Now, regarding acceptable linearity, what we recommend is that the linearity of the drug release across all time points should be calculated and reported for each diffusion cell and compared within and across all IVRT runs. So please do not just calculate or demonstrate linearity for the average IVRT profiles, making sure that you're measuring that and report that for each individual cell release profiles. And while historically the R-square value of 0.9 or more than 0.9 was recommended for acceptable linearity, the truth is, based on all the data that we have seen, the release rate in order to be considered suitably linear should have R-square value of equal to 0.97 or more than 0.97 across the IVRT duration. And in order to better illustrate why we now recommended higher R-square value is really because R-square may not be a very sensitive criterion to show that you are have an suitably linear profile. So for example, this is a release profile, and when we plot the data, we see that we have some curvature and deviation from the steady state release kinetics, although the R-square is 0.969 or nearly 0.97. Again, here at the last time points and the middle time point, last time point and middle time point, we are deviating from linear regression line and steady state kinetics, but R-square value is 0.96. And in the last example, the last time point clearly is not a steady state. Even though the rest of the profile is suitably linear, this time point has a different release rate. And if you observe such a thing, that maybe either you're not at the same condition anymore, or your dose depletion is high, so you need to optimize your method. Or if you have adequate duration of the IVRT and sampling time points up to this point, you can maybe just exclude the last time point. But again, we see that R-square is 0.96, even though we don't have steady state release kinetics for all our samples. The duration of the IVRT study is typically 4 to 6 hours, and duration of less than 4 hours may be insufficient to assess whether the release rate represents the steady state drug release kinetics or not. And for some of the products that the release is very slow, like some of the ointments, you may need to run a longer IVRT study, but typically 4 to 6 hours would be sufficient to capture enough sampling time points during the steady state kinetics. And if you select a shorter duration, like less than 4 hours, which is not recommended, you may need to justify it by compelling experimental data and demonstrate that you try to optimize other method parameters to be able to run a longer duration of IVRT study, and was unsuccessful. That's why you had to select duration of less than 4 hours. And those need to be submitted in your method development report. So the dosing that is used for the IVRT study is a pseudo-infinite dose. You need to make sure the dose is sufficient enough to cover the whole exposed area of the membrane and sufficient enough to provide steady state release kinetics during the IVRT. And while there is no specific range to recommend for the pseudo-infinite dose, your release profile and the dose depletion value would speak to whether your dose was sufficient and acceptable or not. And the dosing procedures that you use should be appropriate for the equipment that you selected for your IVRT study, and you can refer to USP chapter 1724 to see what dosing procedure is recommended for each type of apparatus. But for most of the vertical diffusion cells, you need to have the membrane first equilibrated with the receptor solution and apply the dose on the membrane while it is mounted on a diffusion cell and is equilibrated with the receptor solution. You need to be aware that sometimes the dose application tool that you're using can impact on product microstructure. So for example, if you're using a syringe to apply a dose, make sure the shear force that the product is experiencing while being dispensed from the syringe is not impacting its microstructure. And if the microstructure is changing following dispense of the product, it may change drug release and bioavailability. And another note is that to make sure your diffusion cell is occluded during the IVRT study, because the per Higuchi kinetics, the steady state kinetics, is obtained when the dose is occluded during the IVRT study. Another consideration for the IVRT is the dose depletion, which is expressed as a percentage of the amount of drug in the applied dose and can be reported as an average dose depletion for the entire IVRT study. And per Higuchi kinetics, a steady state release is assumed when dose depletion is less than 30%. But does it mean if your IVRT study has dose depletion of higher than 30%, it's not valid? Well, not necessarily, because for some topical products, steady state release kinetics may continue to be observed at higher percentage dose depletion. And if your IVRT method result in a dose depletion of higher than 30%, it may be acceptable if the release rate remains suitably linear across the duration of the IVRT and you have adequate duration of the IVRT. However, if your IVRT is too short, for example, less than 4 hours, and you have high dose depletion, you need to try to optimize some other method parameters to resolve this. Precision and reproducibility are another part of IVRT validation, and the intra-run and inter-run precision and reproducibility may be compared for the release rate calculated for each diffusion cell. And while a minimum intra-run and inter-run percentage CV of less or equal to 15% is recommended, for best practices we encourage you to target percentage CV of less than 10%. And you need to make sure that a minimum of 3 independent IVRT runs for precision and reproducibility are needed in the IVRT validation. One of the important aspects of the IVRT validation is to establish the discriminatory capability of the method, and the first test to do that is the sensitivity study. Here, you would compare the release rate from the nominal reference strength formulation with that from 2 comparable formulations, one at a higher strength, which is 150% of the nominal strength, and a lower strength formulation that has 50% of the nominal strength. And here are 2 examples of the IVRT sensitivity. On the left, we are showing a sensitivity study which may not be acceptable. And one reason is it was done with a 200% strength instead of 150% strength. And our recommendation is for the higher strength, do not use any strength above 150%. And another issue with this IVRT sensitivity is that there is high variability in the data, and you can see that the standard deviations are overlapping. But on the sensitivity study in the right image, you can see that the 100% strength is well separated from the 50% strength and 150% strength. And this is an example of the sensitivity study that we would like to see in the ANDA applications. If you try to formulate a higher strength and you run into challenges, for example, you observe precipitation of the active ingredients and you think that it may not be feasible to formulate it without substantial reformulations, well, you can use other strategies, but we recommend that you reach out to the agency through control correspondence or pre-ANDA pathways. Another test for establishing IVRT discrimination is the selectivity. Here you need to show that the release rate of the test or reference standard at the nominal reference strength are non-equivalent compared to the lower strength, which is 50%, and the higher strength, which is 150%. And for this, you need to run a statistical analysis test that is recommended in the USP Chapter 1724. While the sensitivity study you need to establish based on visual assessment, here we need actual statistical analysis to establish selectivity. And you need to run 6 cells of each strength and 6 cells of nominal strength of the RS with the 100% strength can be compared with the 6 cells of altered strength 50% and 150, and all 12 cells that are compared should have been run in parallel on the same day. In addition to the selectivity, you also need to establish supplemental selectivity. For this study, you would need to use an altered formulation that has the same API at the same strength as in your test and reference product, but with either altered composition or altered manufacturing process. And you need to show that the release rate of this altered formulation is not equivalent to the release rate from your test or reference product. I would like to note that the altered formulation may include changes in inactive ingredients, type or amount, or even in changes in the manufacturing process or combination of them, because sometimes by a simple Q1 change or Q2 change you may not be able to have a significant different release rate of the altered formulation. Therefore, you may need to alter a few things regarding the formulations. And please note that not all of those variations will necessarily produce a different release rate. And last but not least is the specificity test. You need to show that the IVRT method is proportionally linear to its response to differences in the release rate. So the release rate that you obtained from your selectivity study can be plotted against the concentrations, and you would calculate the R-square value to evaluate the linearity of this regression line. And our recommendation is that you target a minimum R-square value of equal or more than 0.95. Again, historically, the linearity for specificity study was recommended as more than 0.9, but our current recommendation is to establish linearity of more or equal to 0.95 for the specificity study. Another thing that I would like to note is regarding the slope of the specificity curve. So here we have two examples of a specificity study, and in both cases the R-square value is 0.99 and acceptable. However, in the right picture, we see that the slope is very shallow, and the reference strength is hardly discriminated from the higher or lower strength. Especially if you have a little bit more of standard deviation, you may not be able to pass the selectivity study. But in this example, where the slope is higher, the higher and lower strengths are well separated from the reference strength, and definitely this IVRT method is more discriminating than this IVRT method. IVRT robustness is also need to be evaluated as part of IVRT validation, and for establishing robustness of your IVRT, you need to evaluate a few tests. One is temperature variations, and show that, for example, by small variation in temperature plus minus 1 degree relative to your target temperature, for example 32, your release rate does not significantly change, or variation in the dose volume plus minus 10% of the dose amount, or small variations in the receptor solutions, for example changing the pH of the solution, or slight changes in the composition of the receptor solution, and eventually mixing rate variation if the apparatus allows for that. So while you're validating your IVRT method, as I mentioned during the IVRT method development, you need to validate your analytical method that you would use to analyze the receptor solution samples. And the IVRT validation and pivotal studies should use a validated analytical method for the receptor solution samples. It's important to note that you need to submit a separate report for validation of your analytical method and a separate report for the validation of your IVRT, and a separate report for the IVRT method development. And please note that the validation of The analytical method should be performed using chromatography software with audit trails and should include a multi-point calibration curve. So it may not be acceptable to use a single point calibration for validating the analytical method for the IVRT. And finally, the last stage is the IVRT pivotal study, where the drug released from the designated RS and the test products are evaluated, and this is considered a bioequivalence study, and the recommendations related to sample retention would be applicable to this study. A single batch of the test and reference product are compared, and this batch, the batch of the test and reference that you're using in the IVRT pivotal study should be among the ones for which the Q3 properties were characterized. It is recommended that you use a blinding procedure to blind the investigator conducting the IVRT study, and once blinded, then the test and reference product can receive random designation of, let's say, A and B, and they can be dosed on the membrane using an alternative pattern. For example, if the first set of cells are dosed as AB, 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 the second set can be dosed in a sequence of BA, BA, BA. And finally, after sampling the receptor solution at the designated time points, then the release rates for the test and reference product are compared using a statistical analysis called Wilcoxon-Ransom test that is recommended in the USP Chapter 1724, and the equivalency of the drug release between the test and reference product is established. Here are the references for my today's presentation. In addition to USB Chapter 1724 and relevant FDA guidances, there is also a link to the recordings of the presentations and panel discussions that we had last year, an FDA workshop on IVRT and IVPT methods, best practices for scientific considerations for ANDA submissions. And I highly encourage you to use this link and listen to the recordings if you didn't attend the workshop. With that, I would like to thank and acknowledge my colleagues and supervisors at USFDA, especially Dr. Sam Raney and Dr. Priyanka Ghosh. And I look forward to the Q&A session where we will continue this discussion and further discuss and talk about considerations on best practices for IVRT study and answer your questions. Thank you very much.
Thank you for that great presentation, Dr. Ramezanli. We will now take a quick 5-minute break to prepare for our question and answer panel. If you haven't had the chance to enter your question into the Q&A chat pod, please do so now. And in the meantime, enjoy the break.
The only change should be to your API amount. And I don't know why that particular person who asked this needed to introduce Q1, Q2 changes. If, for example, as I mentioned in the presentation, if you have difficulty formulating a higher strength, you can approach the agency or ask question whether you can use alternative strategies. But if you don't have difficulty formulating higher or lower strength, we won't recommend that you introduce Q1 changes and other Q2 changes except for the one inactive ingredient.
Sam, would you like to add anything?
Yeah, I think there's actually two ways that you can make the higher and lower strength formulations, two strategies. One is that you take an ingredient that is, as as Tannaz says, the diluent, really that largest ingredient in the formulation, and you cut off a little bit of that to make room for the additional drug, or you just balance that if you're having a lower amount of drug. So that's one way to do it. And so that naturally would be a non-Q2 formulation typically, and that's to be expected. No issue there. The other strategy is that you take the entire formulation blend that contains all of the ingredients in their native proportions that you have in the reference strength product, and then you take that blend and you basically just have, let's say it's a 5% drug you're adding. You have 95% of your blend and 5% drug, or 85% of your blend and 15% drug. Either way, the point is to minimize the impact on the product and really to focus on changing the strength of the product. And I'll leave it at that because there may be other questions that come up related to this, but I just wanted to clarify that.
Thank you. Thank you, Tannaz and Sam. I think a related question that we received is, can the formulation for the method, can the formulation for the method validation studies, especially I think it refers to supplemental selectivity, be an alternate dosage form? And Hiren, would you like to address this question?
Absolutely. Thank you, Priyanka. So in my opinion, to use alternate formulation, you can use different strategies. So you can use the different Q1, Q2, even changing the manufacturing, and even the change in the ultra dosage form, which you asked, gel versus cream. It should be okay to use the different dosage form to support the supplemental selectivity or to conduct the supplemental selectivity study. Thank you.
Thank you, Hiren. The next question, Hiren, I'm also going to direct towards you. And the question basically speaks to, is it necessary to go to the summary basis of approval for the reference product, for example, and if an IVRT method is available in there, is it necessary to utilize this method, especially if that method suggests that we see 80-90% drug release during the first hour? And how much drug should really be released should be observed in the IVRT studies that we are recommending as part of the characterization-based approaches?
So yeah, I'll try to split the answer. So I think the first part was, is it necessary to go with the SPOM method for the RS product to conduct the IVRT study? It is not necessary to go with the SPOM method. This is your trace product. You can develop your taste product, which is fit with the the equipment that you have it in the lab. So it is not necessary to go with the SPOM method. In terms of if one see the 80 or 90% dose depletion in the first hour, I would suggest to change, optimize the IVRT method in terms of the selection of equipment. They can change the dose amount or, different membranes. So they can use different strategy to minimize that one. As of now, I haven't seen where, 80 or 90% dose depleted in 1 hour. But I would say, try to optimize the method, and if you see any challenges, I would encourage them to submit the pre-ANDA platform and seek the agency's feedback on that one. In terms of the minimum dose depletion amount, we don't have any minimum requirement. The dose depletion should be less or minimum this much, or any restrictions. So as per the Higuchi equation, there was one assumption that the dose depletion should not exceed the 30%, but that 30% is, in my opinion, is very, very arbitrary number. So we don't stick with that. The dose depletion should not go beyond 30%. Even in fact of the application, sometimes we have seen that applicant proposed to reduce the study duration based on the explanation that dose duration exceed 30%. So they were proposing 2 or 3 hour studies. And as Dr. Ramezanli mentioned that, dose depletion should not be considered to decrease the study duration. As long as the linearity is maintained over the study duration, it is okay to go above 30%, up to 50, 60%. It should not be a concern to have higher dose depletion than the 30%. I hope that answers your question. Thank you.
Thank you, Hiren. I think the next question we have is for Dr. Nair. And the question is, what guidelines should one follow for the analytical method validation that are submitted as part of these IVRT studies? I think it mentions ICH or M9 in there. I can hear you, but it's a little bit on the lower side.
How about now? Better? Okay.
Thank you.
Yeah, thank you for that question. Actually, yeah, the analytical method validation, the short answer is, I believe we recommend the M10 analytical method validation. That is the latest. You're supposed to use the most recent version of the FDA guidance on this analytical method validation guidance. And right now, I believe that is the M10. So that's the short answer. Thank you.
Thank you, Dr. Nair. I think a follow-up question that we received related to the analytical method validation is something that was noted on the slide, that it may not be optimum to use a single point calibration method to support the IVRT studies. The question is, why can we not use a single point method if we know that the method is linear? So maybe, Sam, would you like to start responding to that question, and we can go around the panel?
Sure. So I appreciate the question. LCUV, diode detectors, it's traditionally very linear response, right? So I appreciate why you might feel like, well, a single point is going to be suitable. Now, when you're referring to saying that if you know that it's linear, the question is, UV response is linear until it's not. And typically at the lower end, eventually you're going to have some kind of an issue where you don't have a linear and proportional response. And so the problem is that IVRT studies in this context are being used as bioequivalence studies. And we require them to be accurate, sensitive, and reproducible. But we cannot know that they're accurate if the analytical method and the accuracy of the analytical method is unknown. And so while there would be a region within which the UV response is quite linear, you don't know what that point is when the analyte that you're measuring is at a concentration where it's no longer in that linear range. And if you only have a single point, you just never have an assessment of that. So this is part of the reason why, as part of the analytical method validation, as we do for all bioequivalence studies, we want to make sure that there's a multi-point calibration curve to ensure that all sample concentrations being measured are within that linear response range for the analytical method, and that this is done in the same manner as the accuracy and reproducibility of the analytical method is validated for all bioequivalent studies. And so this is the reason why a multi-point validation, a calibration curve is recommended.
Thank you, Sam. The next question that we have is for Dr. Lee. Dr. Lee, what are the agency's suggestions on the raw data submission requirements for an IVRT study?
Hi, can you hear me?
Yes.
So
We cannot hear you anymore, Xiran. I'm not sure what happened there. No. Maybe we'll come back to you in a second and we can check your microphone in the meantime. Would anybody else like to take a stab at that question in the meantime? Hiren or Anil?
Expect the applicant to submit all concentration data for the IVRT validation study as well as the pivotal study, along with the calculation for the release rate and as well as the statistical analysis calculation. So that's what we expect the applicant to submit in the submission. Thank you.
Thank you, Hiren.
Raw data as much as they could with the proper, what are all these raw data for, and what are all the methods that parameters that they use to, generate the raw data. That will definitely help us in review process. Thank you.
Thank you.
Reconstruct the calculations that upon which you're relying to support the demonstration of bioequivalence.
Thank you. Thanks, Hiren. Thanks, Anil. Thanks, Sam. Our next question to you, Tannaz. Are there any recommendations for selecting a membrane for an IVRT study? Do we have recommendations for membranes that should be used over ex vivo skin, or should they be used in unison? So I think there is a little bit of confusion about what kind of membrane we typically use in an IVRT study.
Sure. So for the IVRT study, we are using synthetic membranes, and the most commonly used ones are Teflon, PVDF, or even cellulose acetate, and they have to be inert, and you need to make sure the drug is not binded. And depending on physical chemical properties of your API, you can select the more hydrophobic or lipophilic. We definitely don't recommend using membrane that have lipid components in it, like stratum. They are not recommended for conducting IVRT studies. And another consideration is with their pore size, that in most cases you can target a pore size of about half a micron. Sam, do you have anything to add?
Because they are chemically stable with a variety of receptor solutions. They tend to have low binding for the drug of interest. And what you want to make sure you're measuring is the native steady state kinetics of the dosage form that is not interfered with by any rate limiting diffusion across the membrane. And so that's why you essentially want just a single, simple monolayer membrane that's a support, and these filter membranes that are recommended in USB 1724 are well established to be suitable for a wide variety of IVRT methods. Other types of membranes could have issues impacting the kinetics of and really invalidating the test.
Thank you, Tannaz and Sam. Sam, the next question we have is, I think, directed to both, actually, Tannaz and Nair. And it relates to, I'm missing Hiren. It relates to the fact that somebody noted that on the slides we mentioned that the method validation studies, especially, say, for example, the sensitivity or selectivity studies, need to be run in parallel. And the question is, USP 1724 says that T and R products should be alternately sequenced during the run of the study. So it was not clear to the person how we can run studies in parallel. There is a related question. Someone noted that they just have 6 cells. So how should they proceed in that scenario to run parallel studies?
The recommendation and the compendial method is that the products are dosed, in any given set of diffusion cells, that you would dose, A, B, A, B, A, B. So it's dosed in that alternating sequence, but they're all run in parallel at the same time. And that's essential because uh, the you're comparing the release rates, so you want all conditions of the test to be identical for the test and reference product. This is part of the reason why the sequence of dosing is that alternating sequence, even just to make sure that you don't have effects where the first half of the cells were treated differently than the next half of the cells because of timing or because of other considerations. So the being run in parallel is very important for that comparability. Um, the the second part of it, if you only have a 6 diffusion cell assembly, um, I think one of the challenges in that situation becomes that you've got to be running a lot of tests across different days. And so it becomes essential to demonstrate what the reproducibility of your method is. And I think this can get, then a substantial, because the burden then becomes on you. You've got the additional demonstration that you've not only going to be able to show that test and reference are comparable, but that test and reference, which have been broken up now across multiple days, are reproducible and that you don't have some sort of exaggerated intra-run variability where, let's say, in your set of 6, you've got 3 test and 3 reference run on one day. Now you've got another 3 test and another 3 reference run on another day. And now you're going to combine the 3 test from day 1 with the 3 test for day 2 to get your 6. And that intra-run variability may be greater than the intra-run variability if you just ran them at the same time. These all become sort of the characterizations of the system that you then need to be able to help characterize. But I guess it is feasible to do. It's just that you'd have to be able to demonstrate that reproducibility. Ultimately, also, when you're looking at the nominal strength versus the higher strength, you'd still have to be able to do the comparison of those directly in parallel. So nominal strength versus, let's say, 100% strength versus 150, and get that entire set done. And then independently, you couldn't just do the 50% strength on its own. Again, you'd have to run in parallel the 100% strength versus the 50% strength and get those done in parallel. So it's a lot of runs. It might be worth just investing in a few more diffusion cells.
Thank you, Sam. We are almost out of time. One last question for Dr. Lee before we wrap up. So Dr. Lee, the question is, can the drug solubility that is done as part of IVRT method development and validation be assessed at ambient temperature, or would the agency expect to see solubility data at 32 degrees centigrade or 37 degrees centigrade as relevant for the dosage form or route of administration?
For the question. I hope you can hear me now.
Yes, we can.
Oh, sorry about that. So as stated in the presentation and also in our guidance, the IVRT release rate, the experiment should be carried out at 32 minus plus 1, and the membrane surface temperature should be justified or verified to be stable at 32 before dosing, except the case of the vaginal drug product that for which the temperature should be kept at around 37. So we would prefer to evaluate the drug solubility in the receptor median under the study condition, which is we prefer at 32 or 37, depending on the drug product. Thank you.
Thank you, Dr. Lee. Thank you so much to Dr. Ramezanli for the wonderful presentation, and to all of our panelists for the engaging Q&A session, and to all of you for sending such wonderful questions.